Hi there, in this video we are going to discuss glycolipids. Glycolipids. So what are glycolipids? Glycolipids are basically combination of carbohydrates and lipids. So the carbohydrates present here in the structure of glycolipids can be monosaccharide, can be disaccharide, can be oligosaccharide, or they can be carbohydrate derivative, for example N-styrenic acid and so on. And lipids involved basically include ceramide. So what was ceramide? Ceramide was basically a combination of the sphingosine. Here was a fatty acid and here was C double bond C CH2 12 CH3 and here was CH2 OH. So this was the this was the structure of ceramide. So lipid backbone involved ceramide, and ceramide is basically combination of fatty acid plus sphingosine. So due to presence of sphingosine, these glycolipids are also called glycosphingolipids. They are called glycosphingolipids. Okay. So what is the function of these glycosphingolipids? These are involved in the these are present in the membranes and regulate the structure of membranes. They are present in the outer portion of cellular membranes and regulate the structure of membrane. Moreover, they act as antigens. For example, they act as a blood group antigen. Moreover, they act as antigen of fetal cells and play their role in the development. Moreover, they also act as cell surface receptors. And play their role in the cellular coordination and signal transduction. So these are the different roles of glycosphingolipids. Now we come towards the structure of glycosphingolipids. So depending upon their structures, these glycosphingolipids can be either neutral or they can be acidic. Acidic are negatively charged while neutral are neutral. So in the neutral glycosphingolipids, this ceramide which is the backbone of glycosphingolipid is attached by glycosidic bond to some monosaccharide. So if it is attached to some monosaccharide, then the structure formed is called cerobroside. If it is attached to some monosaccharide, structure is called cerobroside. This monosaccharide can be glucose or it can be galactose. So if glucose is present, it is called glucocerobroside. If galactose is present, then it is called galactocerobroside. So depending upon glucose or galactose, it can be glucocerobroside or galactocerobroside. However, if a disaccharide is present here, if a disaccharide or oligosaccharide is present here, for example, if glucose and galactose both are present here, which is a disaccharide, then it is called globoside. So it can be cerobroside or globoside, and it is neutral, and it is found in the central nervous system and peripheral nervous system and plays its role in myelin sheath there. So it is the basic overview of neutral glycosphingolipids or glycolipids. Next we come towards acidic glycolipids or glycosphingolipids. So acidic glycosphingolipids can be either gangliosides or they can be sulfatides. So what are gangliosides? Ganglioside basically have N-acetyl nuraminic acid in their structure. N-acetyl nuraminic acid is attached to their structural backbone, and these are principally found into the ganglion cell in the CNS. These are found in the ganglion cells in the CNS and play their role there. So, depending upon the molecule of N-acetyl-neuramininic acid, ganglioside can be GM if there is one neuramininic acid, or mono, or D if there are two molecules, di if or T if there are three molecules of N-acetyl-neuramininic acid, or quaternary if there are four molecules of N-acetyl-neuramininic acid. So, depending upon molecule of N-acetyl-neuramininic acid, here the other letter is added. We show the number of N-acetyl-neuramininic acid. So, N-acetyl-neuramininic acid imparts domestic characteristics, and they are involved in the pathophysiology of certain diseases. So, what is N-acetyl-neuramininic acid? N-acetyl-neuramininic acid is also a sugar derivative. It has a carboxylic group here, COOH, then it has CHOH, CHOH, and CH2OH. Three carbons are here. Then here it has a nitrogen atom which is attached to acetyl group, C double bond oxygen and CH3. This is an acetyl-neuramininic acid which is present in the gangliosides. So, these gangliosides are of great interest due to their involvement in certain diseases. However, we need not to go into much details here in this lecture. Next we come towards what are sulfatides. Second class here of acidic glycosphingolipids, sulfatides. In sulfatides, to the basic structure of uh, glycosphingolipid, sulfates are attached, or SO4 group is attached, and it can also be associated with phosphate group or PO4 group. So, this sulfate or phosphate group imparts it a negatively charged structure. So, these sulfatides are principally present in the nerve tissues and kidneys, and they play their physiological role there. So, it was all about glycolipids we should be knowing, and we did not to go into much details here in this lecture. Thank you.